Sorry. Good morning. Good
Oh,
morning from California.
oh, hi, hi.
Forgot. Thought it was later today.
Okay,
Thanks for sending the email to remind me.
I'm so glad that you joined us.
Yes, yes.
We are so happy. So,
What
how
time is
are
it
you?
there? It's late afternoon in Turkey. See, nine.
Oh, I
Uh,
think
6 o'clock.
my internet is bad.
Oh,
Okay, yeah. So, how are you, Mr. Schekman? Are you okay?
I'm fine. Yes, yes.
Okay, so I would like to start with an introduction, so then we can move on to the meeting. Is it okay
Yeah,
for you?
of course.
Okay, now I'm starting. We are the first IGEM team of Koç University. Our main goal is build a biology culture in our university from zero. To achieve our goal, we have started this academic talks concept and a few others. In this concept, we are having guests that we admire their scientific approach. Today, we have a really special guest. Our guest, Professor Wayne Schekman, is an American cell biologist. His molecular biology journey officially started at UCLA in 1967. Even before college, he was interested in biology. In high school, he attended science fairs with the mentorship of his high school biology teacher, Jack Hoskins. You can see Mr. Schekman's first microscope at the Nobel Museum. After the UCLA years, he went to Stanford and continued his education there for 4 more years. After finishing his PhD, he started this long journey at the University of California, Berkeley. The Schekman lab has made a huge contribution to our understanding of vesicular transport, membrane assembly, and membrane fusion. For the groundbreaking work that his lab did, he shared the Nobel Prize for Physiology 2013. Today, yes, UC Berkeley. He's an investigator at Harvard Hughes Institute and the scientific director of ASAP. We are honored and thrilled to have you with us. Thank you for joining, Mr. Schekman. Do you have anything to say? Or if you don't have anything to say, I can move on to the questions.
No, I'm happy to be with you to chat. Happy to answer any questions. Many years ago, my wife, my then wife, and I took a 3-week trip to Turkey, and we traveled around your
Oh,
beautiful
really?
country. Yeah, we visited a number of archaeologic sites. One that stood out was our visit to Ephesus, which is quite a
Oh,
remarkable,
yeah.
find and very well preserved. We also greatly enjoyed a boat ride along the Bosphorus in Istanbul, and we enjoyed touring the city and seeing many of the beautiful sights, and including Hagia Sophia. We visited that. Quite a remarkable mixture of cultures and religions in one place. So it was a very memorable trip. Unfortunately, my wife
And
died several years ago, but that was one of our fond memories.
yeah, it's a good memory. We would love to see you again in our country, in our university, if you want to join us. How can we say no? It's
Yeah,
impossible.
when
So,
when when travel becomes possible again, I'm sure I'll visit.
totally, totally. Okay, so if it's okay for you, I'm going to start with my first question,
Mhm.
and you can see what's going on. It is fair to say that you're a legend in biology, and what I noticed is that in your biology classes, you are giving the names of your students, like he did this or she did this, but in textbooks we will see the Wendy Schekman and his colleagues phrase.
Mhm. Mhm.
Does this bother you in any way?
Yeah, but I can understand why people who write textbooks can't have too many names. I was on the other side of that equation when I was a graduate student with Arthur Kornberg, who was very famous. And one time in giving a talk to my department when I was a graduate student, I referred to our group as Arthur Kornberg et al. And I told the audience that my name was Et, and my postdoc friend his name was Al, Et and Al. So you're kind of used to that.
Okay. Yeah, that's a fair answer. But I have another question. I have lots of questions. When you had your molecular biology degree in USLA, the year was 1971. Now almost 50 years have passed. Can you talk about this journey a little bit? How was the approach to molecular biology then, and how it is now?
Yeah. Well, the approach to solving a biological problem, the sort of what was called molecular biology, was very exciting to me, very new, different than the traditional biochemical approach. It involved using genetics and an emerging technology and cloning genes, and then the ability to sequence DNA and to predict the protein sequence of a gene. It was all very powerful, and I embraced it when I was learning about how bacterial viruses replicate when I was doing research as an undergraduate. But I, at some level, appreciated that to really understand how these genes work in complicated processes, that the traditional approach in molecular biology was not enough. And I'll give you an example. When you have a bunch of genes, such as we discovered when I began my work at Berkeley, yes, you can clone the genes and you can sequence the genes and you can predict the protein sequences, but that doesn't tell you what the proteins do. Just because you can read the amino acid sequence of a new gene product doesn't necessarily tell you what the protein does. It doesn't tell you how it folds and what reactions it may catalyze. So at some level, I appreciated that it would be important for me to learn a more traditional biochemical approach, where you try to identify the gene products by a functional assay, very important, a functional assay, which requires breaking the cells open and isolating the components with a biochemical assay. So that's why I chose to go to Stanford, and particularly to work with Kornberg, because he was a master of biochemistry, and he actually had a kind of dismissive attitude about molecular biology. He thought it was a lazy approach to science. He thought that a tough guy who spent all of his time in the cold room purifying enzymes, and he was a tough taskmaster, but he was really quite a brilliant tactician. He had an intrinsic faith in the ability of biochemistry to solve complex biological problems. And so I learned that lesson from him. But he too had a blind spot. He was so wedded to the idea of studying pure enzymes that he did not embrace genetics. And so when I started graduate school, the year was 1970, his enzyme, DNA polymerase, the enzyme for which he won the Nobel Prize in 1959, had recently, a very important discovery had been made, where a fellow in New York at Cold Spring Harbor had isolated an E. coli mutant with a defect in the gene for DNA polymerase. And these mutant cells grew perfectly normally. That is, they replicated their chromosomes perfectly normally, even though the cells didn't have any of the Kornberg enzyme, DNA polymerase. And subsequently, the other relevant polymerase was discovered. Actually, it was discovered by Kornberg's son, Tom Kornberg. That was quite a remarkable turn of events. And that lesson was very important for me. It sort of imprinted on me, and it said, the lesson I took from that is that genetics is great, biochemistry is great, but the two of them together are even stronger. And so that's what the lesson that I took with me when I started my own program at Berkeley, which was to study this new area or newly emerging area of cell biology, and to develop an approach that would allow me to couple genetics and biochemistry in equal measure, and that worked out very well. And so I was convinced that that was really the way to study problems that could be solved with single cells. And so we used a simple eukaryote, Baker's yeast, to solve a problem that basically applies to all higher organisms. And I still believe that even with the advent of more sophisticated cloning techniques and techniques like CRISPR-Cas9 and all these very remarkable tools, I still believe that in the end, if you want to understand a process at a fundamental level, you need to be able to isolate the molecules and study them and to understand how they interact in a functional way, and not just by running an SDS gel and looking at a polypeptide band on a gel. That doesn't give you all the information that, at least that I would have found satisfying. So the molecular biology has changed quite dramatically. The tools are incredible. When I was a graduate student and I was working on biochemical approach to DNA replication, cloning had not yet been developed. And so it wasn't possible to have these tools to isolate proteins using recombinant technology. You had to isolate proteins by trial and error fractionation using traditional techniques of protein separation. And now you can clone for a pathway where you know the genes. You can clone the gene. You can put a tag on the gene. You can isolate the protein under very gentle conditions by an affinity chromatography approach. And that is an enormous advantage in even just using traditional biochemical approaches. You can isolate proteins that are in very tiny quantities in cells by overproducing them and using tags to purify. So the tools of molecular biology have been a great aid to biochemistry. And so nowadays it makes absolutely no sense to not embrace both approaches.
I believe that's a great approach. I have a similar question. It is fair to say that the scientists from your generation have created the molecular biology culture we have today. Next, aside being a legend in this field, how do you see yourself in this growing culture? And
how do I see myself in what? How do I see myself in
this growing culture?
Growing culture. Okay. Well, so I'm now more senior, and as time has gone on, I've become involved in other things surrounding science, in addition to continuing to do research in my laboratory. And I think for me, this has been a natural progression. I think as I've grown older, I've learned other things, I have other skills. And so I've applied them. I spent a lot of time in recent years focusing my interest on questions of open access and scientific publication. I've had expressed very strong views on this subject. And I feel that that's my role as now a more senior citizen in science, to take a lead that is beyond just continuing to do my own science. I mean, I enjoy doing my own science, but I think I owe it to the scientific community to use my skills in a larger context. And this is something very different than what my graduate advisor, Kornberg, did. He was always focused on his own work, and he didn't become distracted by other things. And it's just a difference between the two of us.
You said you're enjoying your work now, but at the beginning, what made you want to pursue a career in science? Like at first, how was it?
Yeah. Well, I started when I was very young, maybe around 12 years old. I didn't have any particular intellectual interest. I was just an average kid. I was a American baseball fan. I would listen to the radio every day, listen to the baseball games. And then at some point, I don't remember this, I got a present, perhaps it was for my birthday, of a small, very small plastic microscope. And I remember when I collected a jar of water from a local still creek, almost dried up riverbed. And I looked at some of the pond scum on top of this water in this toy microscope. And I just had this fascinating view of this amazing world of protozoa swimming around and crawling in my field of view. And to me, that was just a captivating experience, being able to watch as these crawled around. I just couldn't conceive of how this happened. that really, I think, focused my interest on science, and specifically on microbiology. And that led to the story of the microscope that you mentioned. Later, I forced my parents to use my money to buy this student professional microscope, and that was really that was my pride and joy throughout high school. And but there were no academics in my family, no physicians. My father was an engineer. There wasn't really a role model for science. And so when I graduated from high school, my natural thought was that I would become a physician. I mean, what else would I do with an interest in biology? And so when I went to university, my intention was eventually to go to medical school. You know, in the US, unlike in the rest of the world, you go to 4 years of university, and then you have to apply separately to gain admission to a medical school. So I started with the intention of getting good grades and studying hard and being able to go to medical school. But in my very first year, in my freshman chemistry class, we had this really wonderful professor, and I did very well in that class. And as a result, I was placed in an honors section for the last term of that first year. The year was 1967. And the instructor for that special section was a very famous chemist, Willard Libby, who himself had won a Nobel Prize for inventing carbon-14 radio dating. And he actually wasn't a very good teacher, but it was an inspiration to be in his class. And the real special part about that class was every student was assigned to work with a chemistry professor on a little individual research project for just 10 weeks or so. And I was just by chance put in the laboratory of someone who was studying bacterial viruses. And he had me, since I was completely naive, he had me read a book, the first edition of a very famous textbook called The Molecular Biology of the Gene by James Watson. And it was a revolutionary book because Watson had a way of expressing himself that was completely different than traditional biology textbooks. And as a result of just that few-week experience, I realized that my future lay in experimental science. There was no way I was going to medical school. I didn't enjoy being a pre-med. The other students didn't really care about science. All they cared about was getting good grades and being competitive to get to medical school. And that didn't interest me at all. So I had to tell my parents that I wasn't going to have to be a doctor after all. They were quite disappointed, but they got over it. So that's the journey. And from that moment on, I completely focused all of my interest in working in the laboratory. And
that's awesome.
So It was an unusual experience. I think quite different than my other fellow undergraduates, but and one thing that I tell students who are thinking about a career in science, and this is something that I myself didn't really appreciate until later on, you have to be willing. It's not enough to be smart or hardworking. You know, there are lots of young people who are smart and hardworking and who can do well on tests. I mean, I don't mean to discount that. It's good to be intelligent, but to be effective in experimental science or theoretical science, you have to be willing to gamble. You have to be willing to take risks. And you know, when you gamble, usually you lose, and so you have to navigate the question that you're looking at from many different angles, because you're going to always have to try different ways of answering the question, and you have to be willing to do that and not just take a narrow path that you know will succeed, because that just doesn't work. So there are lots of students at the undergraduate level, perhaps your age, who are at Berkeley, and they're very smart. They do well in classes. They can memorize endless lists of facts, and they very often want to come to the laboratory to explore that possibility. But most of them would not do well as an experimental scientist, because it's too risky. It's too risky. And so many of them choose instead to go to medical school, because frankly, it's a more secure choice. You know, you work hard, you study hard, you do well, and you become eventually, you know, comfortable, very comfortable. You have an honored profession, your parents are happy, and you lead a comfortable life, a meaningful life. You can help people, that's great. But it's, except for very few physicians who are themselves doing clinical research, for the most part, it's not that risky. I mean, personally, now in the pandemic, it's risky, but it's not risky in terms of choices. So you have to, at this stage, for you young people, if you're interested in experimental science, you have to not just ask yourself, you have to go into the laboratory and see if the experience of doing experiments that usually fail, if that doesn't deter you. You have to see if you enjoy the challenge of continuously thinking about a problem and trying to refine it and eventually some understanding. That's the challenge.
I think that's a great approach. And I have a question related to that, but not exactly. I'm going to move to some more scientific questions. So you said you have to take risks, and a lot of things go wrong in biology, but we are trying to control it, like we are trying to control cells. I wonder how far we can control cellular transportation, or is it possible to totally control the whole process of transportation?
Well, if you're interested in therapeutic treatments, yes, there are drugs that can block the process. There may even be drugs that would speed the process along. And there are some examples of using cellular transport to try to design therapeutic approaches. And one example that's of interest now is what we're working on in my lab now are extracellular vesicles. These are vesicles that are made inside the cell, and then they are exported outside of the cell. Not just the content, but the actual vesicle is expelled from the cell. And there's a great deal of interest in using these extracellular vesicles as a tool to deliver drugs to cells. So to design a way of getting an important molecule, a protein or or a nucleic acid inside of these vesicles, and then using the isolated vesicles to deliver that to a target cell for a therapeutic approach. And so I serve on several advisory boards for biotech companies that are trying to exploit these extracellular vesicles for therapeutic intervention in cancer or in neurodegenerative disease. So if that's what you mean by control, if you can exploit the process somehow to either control or to intervene, to introduce new molecules, then I think there are good possibilities there.
Oh, right. So another question. We are an iGen team, and this year we are working on a project which is about biolasers. And so, is it possible to modify and change the Golgi packaging mechanism to help it create a resonator structure like silicotane-coated lysosomes, or artificial peroxisomes like vesicles only containing GFP crystals inside the cell?
Yeah, I suppose that's possible. It would be, if this would be manipulating cells grown in the laboratory, it would be more difficult to do this in a whole animal. Is that the approach that you want to do? Do it in cells, or do it in an experimental animal?
In an experimental animal, I think it will be too complicated. So we are starting small cells.
You can deliver things to the peroxisome, and you might be able to develop some biosensor in the peroxisome with some protein that you've engineered in the cell. It's fairly simple to get things into peroxisomes. All you have to do is to put a little localizing signal of several amino acids on the C-terminus of a protein, and it will go into the peroxisome. So you could engineer some kind of a biosensor molecule and put that tag on it, express it in the cell, and get it into the peroxisome.
Thank you.
I think
for the advice.
For the lysosome, you can probably do that too. The trouble is with the lysosome, it's a degraded. It has many hydrolytic enzymes, and so if you put a protein in there, it has to be very stable. Otherwise, it's going to get degraded. With the peroxisome, it's less destructive.
Thanks for the advice again. Does the intercellular transport activity affect cell senescence? And if so, could it be reversed or regressed?
Cellular senescence.
Yeah.
Is that yeah. Well, this, of course, is of particular interest in something that now is a focus of my attention, Parkinson's disease. So I don't know if you read anything about this, but I've become involved now in a major international effort funded by the Sergey Brin Family Foundation. Mr. Brin is a co-founder of Google, so he's enormously wealthy, and he has a family history of Parkinson's disease. His mother has a mutation in the gene that encodes a lysosomal protein, and it encodes a protein that affects lysosomal function, and he has inherited one copy of this mutation, so he's at some risk. So he's now decided to invest a great deal of money in a major basic science effort. And so we are just now selecting teams of investigators around the world who focus on basic science questions of how Parkinson's works. And one area of great interest is why the cells in the brain that make the neurotransmitter dopamine, why they die progressively compared to other cells in the brain of people who have this disease. One idea is that the cells don't die, but they may just senesce. They may go inactive. And so how does that happen? What is the pathway that leads to death or senescence? And if one understands that at a molecular level, is there some way to stop that from happening? Is there some way to block these cells or to resurrect them? Is there some way to reverse the senescence process? So we have a lot of people who are interested in basic questions of that sort. And that's a very hopeful possibility that there are a number of genes that are known in families with Parkinson's disease. And so there are biotech companies that are working on drugs that that change the behavior of these mutant genes and make the cell more normal. So this is if with Parkinson's disease, if you could, as at the first time that someone presents with motor symptoms, if you could intervene with a drug that arrests the progressive loss of dopaminergic neurons, the disease would be treatable, because patients who have the first symptoms, usually it's not so bad, and they can be controlled by giving them another source of dopamine. And if you can then block the death of the remaining cells that make dopamine, these people could live a completely normal life. So being able to block that progressive death is a major goal that I think is quite feasible. And so we have a number of teams around the world who have various ideas about how to go about doing that. So I'm quite hopeful that we'll learn something that will eventually lead to some effective drugs.
We all hope that. And I have a related question to the Parkinson's disease and your work. For most researchers, practical relevance of their research is not so important, so long as they enjoy working on it. We know that you are the scientific director of ASAP, Parkinson's Initiative. Do you care so much about the clinical relevance of the basic research, or are you leading this initiative to accelerate the collaboration of physicians and scientists? Maybe both.
Yeah. Well, for most of my career, all I cared about was understanding how a cell works. I didn't care whether there was anything practical that came of it. But it turned out in my own work on yeast that, in spite of not having any particular practical interest, what we discovered had a actually very direct practical application. When we discovered that yeast cells have a process of secretion that is the same as in mammals, the biotech industry decided to use yeast to produce recombinant proteins of clinical value, like insulin. So insulin to treat diabetics, 1/3 of the world's supply of insulin is made by secretion in yeast. And so what I learned over the years is that, basic science is, of course, that's what interested me, but it also, when you make a fundamental discovery, there are going to be practical applications. And that's the selling point, why governments should continue to invest in basic science. There's no other way to develop these technologies other than through understanding the process at a fundamental level. And so as I've grown older and sort of more engaged in work that could have practical application, I see the continued benefit of basic science. And with just a little bit of focus on a disease like Parkinson's, I think the basic science really will give us new hints about what to do therapeutically. I mean, Mr. Brin, who's the benefactor for this, he's a computer software designer. He knows nothing about experimental science other than that he's a very smart guy. But he himself realized that after, this disease, Parkinson's, was recognized 200 years ago, and in spite of a great deal of work in recent decades, there's still no cure for this disease. And so he realized himself that we just don't understand enough at a basic level to make the kinds of treatments that will be necessary. So he's the one who decided it had to be a basic science effort. And so it is, all of the people that we're looking at are absolutely doing basic science, but they're doing it with a goal of trying to understand this disease. And so I don't think it's I don't think it changes too much of my strong feelings about basic science, other than that sometimes it can be put in a particular direction that maybe have an important clinical goal. The trouble with many governments when they're investing in science is that they are too demanding that all the work have direct relevance. Excuse me a sec, someone's calling me. I'll be right back. Hello? Hi.
Hey, I'm just going to trace up the cable.
Okay, that's fine. I hope you're not going to turn off the power just yet.
Yeah, we're not going to turn it off.
Okay, good. All right, good.
Isn't he turning it off already?
He's not. Okay. We have some work going on in the house. Where were we? Is that what was the? Did I answer your question or? Can't hear you. You're muted.
Oh, sorry, I muted myself.
Yeah.
Okay. I get my answer. So you are a huge supporter of Ocas's. I want to ask you some questions about that.
Mm-hmm.
We believe that it is ridiculous that the publisher will be the owner of my own work, since they have published my paper. Why can't scientists cannot own their own work? Maybe scientists can make money out of papers, just like patents. What do you think about it?
I agree with you. Of course, it is ridiculous. These commercial publishers make vast sums of money. Elsevier in the Netherlands is the largest publisher of scientific literature, and they have consistently fought against open access or against assigning copyright license to the authors of a paper, because they make tremendous profit, and governments have allowed them to have a monopoly, and scientists mostly have been sheep. They've just been willing to go along. These in the life sciences journals like Cell and Nature and the magazine Science, they have created a kind of a culture around the world where the work that's published only in these very selective journals is considered groundbreaking. And young scholars feel if they don't publish in these very exclusive venues that they won't have a successful career. So these magazines have very successfully branded themselves. Their brand name is what draws people to them, and they have almost a monopoly, and it's been very difficult to fight against. And part of what they fight against is open access, because they want to own the literature. They want to be able to make profit over licenses to institutions. And so fortunately, there's many scientists, a growing number, who are fighting back, who realize that we control things, and we've given control to these people, and we want it back. And so I'm particularly proud of my institution, the entire University of California system, which canceled its contract with Elsevier, because Elsevier was refusing to allow our authors, our scholars, to publish open access and to own the copyright. Recently, the MIT has also canceled their contract with Elsevier. So we're fighting back. I have joined, through my effort with this Parkinson's initiative, we have joined forces with a European organization called Coalition S, which has developed something called Plan S. I should hang on a second. I got to get my mask. I'll be right back. Sorry about that. That's okay. Okay, I have a worker in here, so you'll have to bear with me for a while.
Oh,
Yeah, so people who get funding from this Parkinson's effort will have to publish in open access journals. We won't give them the money to publish unless they publish in open access journals. And the Wellcome Trust in Britain is doing the same thing for all of the people that they fund, and that's a huge organization. They will insist on their authors, their scholars publishing in gold standard open access journals. And we're hopeful that the European Research Council, the ERC, will also embrace this view. But we have to change the culture, and that's the problem. There's a culture of exclusivity, and scientists, it turns out, have egos. They are vain. They like to see their name up in lights, and they think publishing in Cell or Nature is a ticket to a secure future, and we have to change that. And that's why I started this journal, eLife, some years ago, because I didn't like the control that was exerted by these commercial outfits, and I wanted to try to do something to make them less influential. And it's a hard battle. It's a hard battle. But in a country like Turkey, and at your institution, most institutions can't afford the license fees for these journals. And Elsevier, they're pirates. They're just pirates. They go around, they negotiate contracts with individual institutions, and they raise the rates. It's just outrageous. University of California canceled a contract with Elsevier now almost 2 years ago. And I helped the university look into some comparison financial figures, and it was really very simple. It cost the University of California $1 per download of a paper in any Elsevier journal. $1 per download. And but what does that compare to? So then I looked at the cost for another single science journal, the Proceedings of the National Academy of Sciences, the PNAS. And that cost per download was only 4 cents. 4 cents per download for a PNAS paper in the whole UC system versus $1 per download. So Elsevier was making a huge profit. And then I found out that Elsevier conducted negotiations with institutions around the world, and they insist that these negotiations be conducted in secret. They don't allow the numbers to get out about what the costs are. The numbers are just all over the place. I was in South America and learned that in Argentina, Elsevier was charging them what amounted to $5 per download for a country that had a science budget that was being cut because of financial the country. And I imagine that in Turkey at your institution, or however those negotiations are conducted, I'm sure that Elsevier is charging way too much. They make a huge profit. And they make a profit on the labor, on the free labor provided by scientists who review the papers or who submit the papers to them. They make money, when you submit the paper, and then they make money when they charge your institution to read the papers. It's a racket. They are pirates. They need to be brought down, and government agencies must exert control.
Yeah, thank you for creating this awareness, and thank you for fighting for us. Yeah, I must say, I believe that you did an amazing jobs. And do you think that publishers will adopt an open access policy someday? Do you think it's possible?
Yes, I'm seeing more and more evidence of it, and the universities who pay these license fees to the publishers have enormous influence. If they simply, like the University of California and now like MIT, if they simply say, we're not paying, we'll just not, our scholars will just not read your magazine. They'll just publish elsewhere. If universities or governments or funding agencies adopt the policy similar to what the Wellcome Trust has done, or this European Coalition S, the publishers will have to change. And we've seen now where a major publisher has finally started to change. Springer Nature has recently agreed to the principles of Coalition S. And so they will be forced over the next few years to make the work in their journals open access. They're still going to make money, but at least the papers will be open access. And so Elsevier, I just don't understand how they can continue to fight. They're in a losing battle. I had the chief executive officer of Elsevier come to visit me at Berkeley some months ago. And I just told her that if they didn't change their policy, I was going to continue to lobby other institutions to cancel their contracts. And she didn't like that, but too bad.
You are doing the right thing, so thank you for that. We have a question in chat. In academia, citations matter a lot. Google sorts articles heavily by citation count, which makes me believe that the articles are not cared about what's inside but popularity. How much is this matters, and does internet inevitably makes popularity a key in academia?
Yeah, this is a problem. For many years, I've argued as forcefully as I can against the use of these measures, like impact factor. About the journal impact factor. This is a number that is calculated that basically is the measure of popularity of a journal. And so journals, as a result of the misuse of this number, have, particularly the commercial journals, they end up making editorial decisions based on what they think will be the popularity of a research article. And I think this is a false measure. Some of the most important discoveries weren't recognized widely until many years later. That's the nature of originality, And so if you rely on something that's going to generate a lot of citations very quickly, you won't necessarily pick the most important science. You'll pick the science that's most popular, but not most important. So I take an extremely dim view of using this impact factor as a measure of anything. I think it's nonsense. But I'm also not so keen on just measuring the total number of citations for an individual paper. It's a measure of popularity. Here's an interesting example. I don't know if you are familiar with this, but for some years, been a lot of interest in circadian rhythm, the clock that cells and organisms use throughout the day to control metabolism. It was known to be a phenomenon in all organisms, even microorganisms. And the first successful approach to understanding the mechanism of the circadian rhythm was done in the fruit fly, Drosophila, by a very famous geneticist named Seymour Benzer, who was a professor at Caltech in Southern California. And he published a paper, probably 50 years ago or so, that where they isolated a Drosophila mutant that had an altered clock. That was the foundational paper. It was published in the PNAS. Absolutely gave birth to a whole field. That paper was cited a total of 10 times over the next 10 years. 10 citations in 10 years. But it was revolutionary. That's why no one recognized it. It was just a curiosity until people, embraced it. And unfortunately, he died before the Nobel Prize was assigned to that work. But he was absolutely the original guy. And if it had been on the basis of citation, wouldn't have even gotten a research grant. So that's very dangerous so how do you measure importance? sometimes the importance of something just takes a long time to understand.
Even the Mendel's work is recognized years later, maybe 50 years or so, something like that. So I believe that citations are important, but not that important.
Yeah.
So we have 2 more questions, but we have 3 minutes. Is it okay for you? Can we ask?
I was late to join you, so I have time.
Okay. So I wonder this because in Turkey, we don't have evolution in our curriculum. So is it possible to understand biology without evolution?
Not really. Why is evolution not taught in Turkey? It's not in opposition to any Muslim beliefs, is it?
But we don't know. I don't know. I believe anyone doesn't know. So it's a thing.
It's just
maybe for that, yeah. What you said is maybe the correct one.
Yeah. It's not against any particular, I mean, there are only sort of fundamentalist positions in Christianity that reject the notion of evolution. You know, so those few people who take the Bible as the literal word of God, word for word, you know, they reject evolution. But as far as I'm aware, the Muslim faith doesn't have that at all. So I don't understand why it would not be taught in your schools. Anyway, it's a shame. Because it is fundamental. It is absolutely fundamental. You can't understand biology without an appreciation of how we got here. You know, it's written in all of our genes. The evolutionary record is there in all of our genes. The genes that we discovered in yeast that are required for protein secretion have the same genes in the human genome. That those genes have been preserved over 2 billion years in evolution. And this is the first I've heard that was not being taught in the schools in Turkey. That's hard to understand. Why do your teachers justify that? Have you asked your teachers why they have?
Of course.
And what
they don't know either.
Well,
they don't know either.
They can't. The teachers can't control the curriculum.
No, the government does all the work. I don't know. In our university, of course, we have evolution, but in high schools and on that level, we don't have evolution in our curriculum.
How do they offer some alternative? Do they talk about?
No. They talk about variation, but they don't talk about evolution. Yeah, that's weird.
That really is hard.
They just skip the issue. They just skip the evolution. They don't talk about creation. They just omit it altogether.
And what if a student asks a question about that in class?
I don't know what's gonna happen. Yeah, I don't know.
It depends on the teacher, I guess.
Yeah, because most of biology teachers don't believe in evolution.
They don't believe in it, but weren't they themselves university students? Didn't they have?
I think
didn't
so.
They have biology at university.
They did. They have to, but they don't believe in evolution.
They don't believe it. So what do they? So then, what is their alternative explanation?
Creation, maybe. I don't know. I really have no idea.
But in the Muslim faith, is there anything like that explain? You know that.
No. There is nothing against evolution, but they still don't believe in it. Yeah, that's weird. I don't get it either. I'm living in Turkey for 19 years, but I have no clue.
Well, that's a deficiency in the educational system then. You know, in the US, of course, this is very controversial, but it's because of this fundamentalist view that, you know, that evolution is against God's word. And so there's active opposition to teaching evolution in certain parts of the country. Not so much in California, although certain schools, but not so much in California, but in the, you know, in the American South. there is a very large proportion of people who are fundamentalist Christians, and so they oppose teaching of evolution. And there's even a museum in Kentucky, in the U.S., a well-funded museum that shows, a model of a dinosaur with a human being on top of it, and kind of rubbish. It's the stupidity is monumental. It's a constant battle. It's a constant battle against these people and trying to keep them from, insisting on textbooks, downplaying evolution, and it'll just never go away.
In Turkey, we are losing the fight right now. It's not that shiny.
I think it must be from the representation of the evolution in Turkey, because when we were kids, our friends always make fun of, we came from monkeys, and it's the representation of evolution. They don't really understand how it works.
I see.
Yeah,
so
that's one of the problems.
So it's just ignorance more than opposition based on some religious views.
Yes.
Yeah. Well, ignorance is pervasive around the world.
Yes.
There's another ignorant strain that's not necessarily the same people who oppose vaccination. And now with the pandemic, so many of us are pinning all our hopes on having a vaccine, but there's a significant fraction of people already in the US who say they won't even take the vaccine because they don't believe in it. They'll believe it's dangerous. And again, it's pervasive ignorance.
Actually, I have a question about that vaccine as well. Is it possible that such a rushed vaccine actually be dangerous?
Yes, of course. That's always a concern. And that's why at first people were so reluctant to say that we would have a vaccine quickly. Because it turns out that with some immune reactions, if the antibody binds to the virus in the wrong way, it could actually exaggerate the infectivity of the virus. Could actually make the virus more potent. So you need, when you have a vaccine trial in this critical phase 3, which isn't just now becoming done for some of the SARS-CoV-2 candidate vaccines, you need to test it out on tens of thousands of people to see if there are some negative effects. And there's a significant possibility that many of the efforts underway now will fail for that reason. They will actually be worse than nothing. I mean, the more likely outcome is that they won't do enough. And the hope is that maybe just a small number will finally make it through and shown to be effective and safe. So safety and efficacy are two absolutely critical conditions. And the worry that some of us have is that our dear leader, Trump, in this country, is so desperate to get reelected that he will, just as he did with hydroxychloroquine, he'll start saying, oh, we should start making this vaccine available even before the testing is finished. And that could be potentially very dangerous. So the hope is that people who are in charge of the vaccine programs will be more open about the data, and that the governments that control the distribution of the vaccine will make sure that it's safe and effective. And then we have to convince people to actually take the vaccine too, or else they're putting themselves in peril.
And not just themselves, but they're putting them. The whole society is in danger.
Yes,
if they're not vaccinated.
Yeah. Well, the people who are most in danger are the fools who refuse to be vaccinated.
Yeah, that's
and unfortunately, they're children whom they refuse to allow to be vaccinated.
That's That's so sad.
You know, we've had
So-
right now, polio is almost extinct, but there continue to be groups, you know, in the Middle East and in Afghanistan and in Pakistan, among the Taliban, who kill healthcare workers who administer the polio vaccine because they think it's some, you know, government control that's, you know, going to sterilize people. It's not actually a vaccine for polio. If it weren't for these violent, resistant fundamentalists, polio would be extinct. It would be like smallpox. It would be gone from the world. There's a chance to completely eradicate the disease, but it keeps cropping up because of this dangerous resistance.
So we have lots of questions, but I think our time is up. So should I finish with one more question? Is it okay for you?
Sure.
Okay. So as last words, what advice can you give for aspiring students who wish to work on molecular and cell biology? You talked about it a bit, but there.
Yeah. Well, for any student, what I advise is that it's very important at your level in university to be able to work in a laboratory and to be able to understand what's involved in doing experiments and, you know, making mistakes and having to redesign the experiment and repeat over and over. That experience is irreplaceable. You can't get that experience by just taking classes and reading textbooks. It's not the same. You have to get into the laboratory and to explore.
Well, in this pandemic, it's not possible. What can we do in our homes?
Well, I don't know what it's like in Turkey now, but in the U.S., many laboratories are beginning to open up. We just last week, my laboratory reopened up to, you know, sort of a low level of work, and it's a bad time. I agree for that. So one thing that you can do that's still obviously very safe is to read the literature, to try to read primary research articles and to understand how the experiments were done and to understand what controls were done to make sure that the evidence is believable. And you can join together in what we do with my research group over this pandemic is we've had journal clubs every week where somebody picks a paper in the recent literature and they review it, and we all take turns criticizing it. So that's something to do. Working in the laboratory, read the literature. One interesting little thing to do is to go to this thing called the bioarchive. Do you know what the bioarchive is? Yeah. So where they post, many people now post their work when it's ready to be submitted to be reviewed, but it's before peer review. So this is a great opportunity for you to take a fresh piece of work that hasn't yet been peer reviewed and do it yourself with a group of students, to go through a paper and find the flaws and see what mistakes might have been made. And I think that's a very good exercise.
Yeah. You'll definitely do that. So thank you for joining us. It's an amazing honor to have you with us.
Remind me again, your university is in which city?
Istanbul.
Oh, you're in Istanbul. Okay. Yeah.
If you want to come to Turkey, come to Istanbul.
Sure.
Yeah. After the pandemic. So we'll be so glad.
I remember when my wife and I traveled to Turkey. Oh, now maybe 8 or 9 years ago. We in the villages. Maybe it was the time of year we were there. There were big posters on the sides of building that showed all the high school graduates, or all the names of all the who won special honors on graduation, posted on big sides of buildings. It was quite impressive. So people obviously are proud of their children. That's good.
are using people for marketing. So
I see. Okay. Well, it wasn't obvious that they were selling anything. It was just, here's our great students that have just graduated.
and here is our great school, or something.
They were advertising the school. I see.
They are advertising their education qualities.
I see. I guess I thought these were the names, as I recall, these were the names of students who were going on to university from the high school.
Yeah.
So that's good.
It's common in there. So thank you for joining us again. It was a great honor to have you.
Let me suggest, if you haven't already done this, I have a young colleague, a Turkish colleague in my Elçin Ünal. Do you know that name? Elçin Ünal.
I don't know, but maybe some of our friends know.
Faculty member in my department. She's a wonderful young scientist. It would be particularly good if you could have such a conversation with her.
I will email her after this meeting immediately.
Okay.
Thank you for the suggestion.
Right. Good luck to you all.
Thank you very much. That we can see you in real life later.
I hope
Thank
so.
you again.
There's much that I did not have a chance to see in Turkey, so
We would love to have you.
I'll be back.
So,
All right.
see you.
Stay safe.
Thank you.
Bye.
Thank you very much.
Yep. Bye.
Çok tatlı bir adam değil miydi?